Dear students, today we can discuss about the process of organogenesis during the development of an organism, how it takes place. The basic steps in developmental biology are formation of gametes called gametogenesis, fusion of gametes called fertilization, then as a result of fertilization, single cell zygote will form. Zygote will undergo repeated division called cleavage in different planes and a blastula is formed. The blastula will transform into gastrula. The process is called gastrulation. During gastrulation, various types of movements of cells take place called morphogenetic movements. Some examples for morphogenetic movements are epiboly, emboli, invagination, involution, delamination, etc. That means the movement of cells to outside, inside and spreading of the cells on the embryo. Last stage of gastrulation, three germ layers will form as a result of movement of the cells. These germ layers are called ectoderm, endoderm and mesoderm. From these primitive germ layers, organs and organ systems developed. So this formation of organ and organ system from the gastrula is called organogenesis. We can see various steps in organogenesis from the following slides with explanation.
Organogenesis is the process by which the ectoderm, endoderm, and mesoderm develop into the internal organs of the organism. Cells of each germ layer, like ectoderm, endoderm, and mesoderm, undergo proliferation, migration, reaggregation, and differentiation into various tissue that forms the organ. This process is called organogenesis. This figure or the chart shows extra embryonic tissues and embryonic tissue fertilized egg. For egg will converted into blastocyst, and blastocyst having the peripheral cells called trophoblast and inner mass of cells are there. The inner mass of cells again having layers. One is a hypoblast and other is the epiblast. Epi means outer layer of cell, hypo means below the epiblast cells. So the hypoblast cells forms extra embryonic endoderm, then converted into yolk sac. The epiblast cells will differentiated into amniotic ectoderm and a primitive ectoderm. The primitive ectoderm directly converted into embryonic ectoderm. Then another way, the primitive ectoderm will form primitive streak. Then from the primitive streak, embryonic mesoderm and embryonic endoderm are formed. These three are the future or the prospective layers for organogenesis: ectoderm, endoderm and mesoderm. The germ layers in organogenesis differ by three processes: fold, splits and condensation. In this slide, you can see what are the future organs formed from ectoderm, mesoderm and endoderm. From ectoderm, epidermis of skin and its derivatives including sweat glands, hair follicles, then epithelial lining of mouth and anus, cornea and lens of eye, nervous system, sensory receptors in epidermis, adrenal medulla, tooth enamel, epithelium of pineal and pituitary glands. From the mesoderm, notochord is derived, skeletal system, muscular system, muscular layer of stomach and intestine, excretory system, circulatory and lymphatic systems, reproductive system except germ cells, dermis of skin, lining of body cavity and adrenal cortex. From the endoderm, epithelial lining of digestive tract, epithelial lining of respiratory system, lining of urethra, urinary bladder and reproductive system, liver, pancreas, thymus, thyroid and parathyroid glands.
From these slides, I think you know the concept of organogenesis. Next, we can see an example of organogenesis is the development of vertebrate eye. The structure of vertebrate eye. Eye having Things called as sclera, choroid, retina, then chambers of the eye are aqueous chamber, vitreous chamber, then iris, pupil, eyelid, nictitating membranes. All these are the parts of the eye. From the following slides, we can discuss how the development of eye take place during the process of organogenesis.
In the process of development of eye, tissues of eye are differentiated from the wall of diencephalon called neural ectoderm. From that it forms a retina, the epidermal ectoderm, the lens is formed from the mesenchyme or the mesoderm of the head. Fibrous and vascular coats like sclera and choroid are formed. A pair of ventrolateral evagination develops from the wall of diencephalon called optic vesicle with a cavity called optocele. The basal part of the optic vesicle is called optic stalk. In this slide, the optic vesicle continuously pushed towards come in contact with the lateral head epidermis and induces a thickening called lens placard. Now, the external wall of the optic vesicle flattens out and begin to invaginate to form a double-walled optic cup. The invagination of the optic vesicle towards ventral side of optic stalk form a double-walled groove called choroid fissure, through which blood vessels supplying retina and other structures enter the optic cup. See in the figure, you can clearly see the optic cup, then inner layer that is called prospective retina, and the outer layer is a prospective pigmented epithelium that we can discuss from the next slides. The outer and inner wall of optocele and the rim of the optic cup form the edges of the pupil. Later, the edges of the pupil become thin and form the iris. Pigmental granules develop from the external thin layer of the optic cup, forms pigmented layer of retina. In the previous figure, this pigmented layer has been shown. In the invaginated thicker inner wall rapidly multiply and form nerve cells, supporting cells, rod cells and cone cells, which are the sensory cells. The nerve cells of the retina give out nerve fibers towards the cavity of optic cup, come out of the eyeball called optic nerve. Diagrams shows lens vesicle, lens capsule, optic stalk, prospective cornea, primary fiber cells. And in the next diagram, lens epithelium, cornea, vitreous body for the vitreous chamber, future optic nerve, pigmented layer of retina, lens fibers, sensory layer of retina. See the location of these structures in the future eye. At the time of formation of the optic cup, the lens placard invaginates and form a cup-like structure called lens cup. The lens cup gradually pinches off called lens vesicle. The cells of the inner wall of the lens vesicle elongate and form lens fibers. Some of these cells divide by mitosis and form more fibers inside lens vesicle, that is obliterated, and a solid lens is formed. Cells forming the lens vesicle undergo cytodifferentiation and form the solid lens. When the lens vesicle is pinched off from the ectoderm, free edges of the outer ectoderm extends over the lens and in conjunction with the head mesenchyme forms a double layered cornea, which modifies into a thin and transparent membrane over the lens. This slide explains the formation of choroid and the sclerotic coat of the eye. The mesenchymal cells accumulating around the eyeball forms a coat of connective tissue around the optic cup. Outside the pigmented layer of retina forms the middle choroid coat. Outside the choroid coat, the mesenchyme cells form a tough fibrous capsule called the sclerotic coat or the sclera, which provide protection to eye and its muscles. Chambers and fluids of the eyes are formed. And there are three chambers of eye: anterior chamber between iris and cornea, posterior chamber lens and iris, vitreous chamber between retina and lens. The fluids of the eye are aqueous humor and vitreous humor. Aqueous humor, posterior chamber it is produced and in the anterior chamber it is drained. The vitreous humor is a gelatinous material derived from cells of retina and lens, maintain the eye shape. Then two eyelids and nictitating membrane develops at a later stage. The inner lining of epithelium of eyelids forms the conjunctiva which is fused with the cornea. This is how the development of eye take place in a vertebrate organism.
Dear students, from this I hope you could understand the concept of organogenesis and the development of vertebrate eye in detail. Thank you for watching. See you in another class.